within host evolution of flu in particular. I guess the reason for doing that in theory is that there's a kind of perspective on viral evolution where you think about a global population and changes within that population. But really that population is comprised of very large numbers of people and individual cases of infection and people giving that virus to each other. So sort of, in a way, the population only exists as a series of individual infections. So just to think about how we can study that, we're going to use a framework of population genetics. Just to give a very basic introduction, we're thinking about variants in a population. So here we have a set of genomes, and we're going to see there's some position in them. There's some individuals with a C nucleotide, some with a G. And so that's what we call a polymorphism. And we can think about the frequency of that variant and how that variant is going to change in frequency over time. And because this is a physics conference, I wanted to put up an equation. This is Kimura's diffusion equation, which if you think of the frequency as a probability distribution, this describes how the frequency is going to change over time. You might be thinking of a Fokker-Planck equation, which is pretty spot on. It's got particular components that you can think of in a more biological way, though. So we have here selection, so a parameter sigma determines whether a variant is, if it's positively selected, it will increase, and so forth. We have mutations, so this is changes from the variant into and out of the particular nucleotide. And then, perhaps confusingly for physicists, we have this term, the diffusion term, is called drift, genetic drift. So we have these three parameters, population size, selection, and mutation rates, which together kind of describe how the system's going to evolve and what's going to dominate over what over time. So using this framework, we can apply this to data. When I started in evolution, we were starting to get data from populations collected over time. So here we'll imagine a variant from a population measured at regular intervals. And we can use an equation to infer what is selection, and how is selection affecting this variant. So here what we do is we have knowledge of the frequency Q over time, and we use that to infer some parameter sigma. So that tells us how strong is selection on a particular variant. So here positive selection in favor of this variant. And given that, we can then start to make predictions. So if we know sigma and we know the frequency at a given time, we can sort of predict ahead what's going to happen, what's going to be the change in frequency with time, and so thereby sort of make predictions about what will happen in the system. Okay, so that sounds quite simple, but the challenge is that viral genomes work as genomes. It's not a competition between alleles, it's which virus wins the fitness competition. And genomes might have lots of different variants on them. So our variant here is not purely in isolation. So while our basic equation describes what's going to happen to the frequency of that due to its own selection, it's also true that that selection will affect other variant frequencies in the genome. Here it's related to this term Dij, which is a statistical measure of the sort of propensity of the variants to appear on the same background or on different backgrounds. So again, it becomes more complicated, but we can think about these kind of linkages between variants and taking those into account, start to do a bit with those. Just to note, in flu, we're thinking about flu segments, so it's 8 segments in the virus. And linkage within segments is almost always preserved. There's not really much recombination going on, whereby segments sort of form hybrid copies of themselves. So we do need to model linkage, but we can sort of do that. And instead of inferring a coefficient, we can infer something of a fitness landscape. So this is data from a hybrid H5N1 virus grown in ferrets. And we've taken data over time and we've learnt what are the underlying sequences. So using variant data to infer these sort of different combinations of loci down at the bottom. And then here, instead of a number, instead of a selection coefficient, we've got fitnesses shown relatively in the color of each of these sequences. So it's kind of fitness landscape describing what's going to happen. And we see here that in this particular experiment, the virus has gained very rapidly in fitness. It's a sort of slightly odd hybrid virus. And in theory, I mean, this was done to look at the emergence of new pandemic viruses. And here we're showing that in a way you can start to do prediction of what's going to happen based on experimental data. You can start to predict how a pandemic might emerge, with the caveat that we've shown only a really very, very tiny part of the overall fitness landscape. So in theory, it would work if you had an infinite number of varied experiments, but it becomes, given the data available, quite a limited sort of prediction. So we can then think, well, what about humans? And this is data from a flu challenge study in people. And what's happened here is that the viruses have been grown in cells, and they pick up variants that confer sort of a signal of cell adaptation. So these 4 variants on the right-hand side help the virus to grow in cells. And then what happens when it gets back into people is you can see you have this sort of network, and the virus makes its way sort of back to this red genotype in the bottom left corner. So what you see is the reversion of cell adaptation in the human. But we don't see any drug resistance. So what's interesting is we have a cohort of people here who were given oseltamivir really from day 1 of the study. And yet we see almost no sign of that whatsoever. In fact, literally no sign of drug resistance turning up over a few days. So then it's a sort of limited period of time seems to limit what can happen. It's really only a few days. And so that, if you like, we see bad mutations disappearing and things that are in the population being wiped out, but not a strong signal of positive selection. So that's thinking about drug resistance. We can also think about immune evasion. And here's some interesting data from a study of swine flu in pigs. Again, an experiment. This was done in the vet school in Cambridge. And what's interesting here is we see this sort of clade in the middle, this CGG variant. So this is having C at positions 447, a G at 553, and a G at 844. And what happens is this clade sort of grows for a bit, and then towards the end of the infection, it dies out. So we see the wiping out of this G variant here, this A to G at the 3rd position. And what's sort of interesting here is the A to G codes for a non-synonymous mutation in an antigenic site in the protein. And so it's potentially a signal of adaptive immunity, wiping out this variant. But what we notice that the adaptive immunity kicks in. This is a model of time-dependent selection, and the antigenic signal kicks in later on in infection. So there's a kind of delay for the host to generate the adaptive immunity against that variant. And that might be pretty important. So again, this is data showing the viral titer. This is from horses. And here we see a sort of pattern whereby there's a very sharp increase in viral load upon infection. You then have an innate immune response, which kind of lowers it by a couple of orders of magnitude. And then the adaptive immune response is what finally clears the infection. And so if the peak in the viral load is very early, but the sort of the effect of adaptive immunity kicks in much later, it's potentially true that the adaptive immunity has very little effect actually on what gets transmitted. Because if the transmission happens here and the selection happens over here, then it hasn't got, the selection isn't sort of on time to be able to change the composition of the virus before it's transmitted to another host. So that's some notes on selection. We can also think about reassortment. And here we said there's no recombination within segments, but what about reassortment between segments? And so again, we looked at this from this is our human challenge study data. And when we looked at this, we did an inference of selection, and we actually infer that the rate of reassortment is very low. So a kind of way to understand this is, here's one of the individuals in the study. And we see different changes in variant frequency over time. And kind of the key thing here is that these 2 variants behave in a very sort of similar way. If you look at this blue model here. So these seem to be doing very similar things as if they're linked together, but they're in different segments in the virus. And that's actually a signal of low reassortment in the host. So within these people, there seems to be almost no reassortment happening between the viruses that are in that host. So why is that happening? We think that tells us something about within-host population structure, that people are not these kind of well-mixed liquid vessels, but actually the viruses are in different places in the host. When viruses get into the airway, they form their own little colonies. And while reassortment happens in cells in each small location, it doesn't happen so much between different parts of the airway. And so we think that spatial structure is then important in limiting reassortment, at least in human infection. Thinking about population structure, we've started looking at data from immunocompromised hosts. And here's a case where we see actually divergence of the virus within the host. So we infer in this individual that we have two clades. There's a clade A, which the virus starts off as being that. And then we see at distinct points over time, signs of another clade, clade B, which sort of gradually moves away from this kind of initial clade. So one lot of viruses stay around these sequences here in sequence space, another one sort of drifts off into other parts of sequence space. And we use that data to look at what is N, what is the effective population size. And if we look at the data, the bit that stays in clade A, we can infer sort of how fast that's evolving. And then fitting a model of genetic drift, we see a very slow rate of evolution once we account for noise and within host structure. And then we can infer a value of N. So we get a very high N of well over 10 million. So that's a sort of rapid summary, which I've tried to compress a bit for time. But just to summarize where I think we're at the moment, if we look at regular infections, we do see selection if there is diversity in between two clades. So a stronger virus will, over time, outcompete a weaker one. But it's likely that the adaptive immune system may have only a relatively small effect. So in essence, by the time that somebody's produced enough B cells to impose selective pressure on the virus, there's not, it's sort of past the point at which transmission is most likely. So it might be that antigenic selection is actually fairly limited on the scale of a single host in a typical infection. But we've shown that if an infection is longer, so for example, in an immunocompromised host, the conditions apply for variants to be able to reach high frequency under selection. Even with relatively weak selection, we would still expect to see anything beneficial coming up to reach higher frequency. And that sort of interestingly matches some ideas that have come up with SARS-CoV-2 and sort of within host stuff in longer infections, potentially generating some of these variants. But in a way, this is an area for future research. It's something I'm still working on and hoping to carry on working on in the future. So to give some thanks, these are some people in my lab and elsewhere who've worked on this, and funding is from various sources. And I'm happy to take questions either now or any time during the day.